Hello everyone, my name is Erik Melén. I'll be giving today's professor's lecture here at Huddinge, Karolinska. My title is Epigenetic and Transcriptomic Signatures of Early Life Exposures, Relevance for Asthma and Allergy. And I'll start a little bit about who I am and what I do in everyday life. I'm a pediatric allergy at Saxka Children's Hospital at Södersjukhuset. But I got my education in med school in Uppsala University and graduated 2001. And a PhD here at Karolinska in epidemiology, followed by postdoc at Harvard Medical School. And since a couple of years, I'm at Saxka Children's Hospital as a pediatric allergist. And I see patients with severe asthma and allergies and lung problems. And here I'd like to present 3 of my patients with respiratory problems. We have Marcus, he's 5 years of age, has a mother with asthma and poor housing conditions. Peter, he's 12 years of age, born prematurely. He had this RSV, respiratory syncytial virus bronchiolitis, first winter, and also lives on a very high traffic street here in Stockholm. Johanna, she's 70 years old, eczema since infancy, and she has now food allergy. So the really question in the clinic for me as a pediatrician, will these patients get a chronic lung disease? And the challenge is, what we know today, is that 10% of children have asthma. So it's a very common disease. We don't have any curative treatment, and we are now concerned about future health. And studies report up to 5 to 10-fold increased risk of later chronic obstructive pulmonary disease. And also the notion that many adult patients with COPD have a childhood origin. So with the patient in front of me who has asthma and may have a severe asthma, it's really on the long-term consequences and what can we do to optimize asthma control and lung health for these particular patients. Longitudinal studies in adults have shown really the tracking of lung function throughout life. And this is a figure explaining the lung function here as forced expiratory volume in 1 second, as a percent of the predicted maximum value. And typically at the age of 20-25, we reach the maximum lung function in our life. On the x-axis here is age, from birth up to age 70 or 80. And if you look at the green line here, that's the optimal lung function curve. With the optimal growth, and you reach your peak height at the age of 20-25, followed by a slow decline. The red line here, these are perhaps smokers who have a rapid decline in their lung function. But now the focus is really on this group as well, who may not reach their optimal, the maximum lung function early in adulthood. And these subjects are at risk of developing a chronic airway disease and chronic obstructive pulmonary disease. So what we are focusing and try to understand is the factors that determine a poor lung function growth. So if we look at the factors and the trajectories from childhood to adulthood when it comes to lung function, again, this is a normal curve that we would like to see. This is the low lung function growth trajectory, and today we'll focus on a couple of those factors, for instance, premature birth, early life environmental exposures, respiratory tract infections, and asthma. The dotted line here represents the catch-up, and I'll get back to that. If you have had a tough start in life, what can you do to get back on track and have an optimized lung function growth? So if we start with preterm birth, and as a pediatrician, I often meet patients who are born prematurely. The definition is if you're born before 37 weeks of completed gestation, you're born preterm. And you can be very preterm or extremely preterm depending on the gestation age you were born in. And this is rather common. 5 to 15% of children worldwide are born prematurely. And what we know when it comes to lung function is really that every week counts. So if this graph shows you the lung function later in life, here expressed as a normal FEV1 by FVC ratio as a measure of airway obstruction. If you're born term, you're here and you have a normal lung function, so to say. For each week shorter gestation, there is a lower lung function eventually when you grow up. And also for subjects who were born just a few weeks earlier, we have shown that actually a couple of % lower lung function is seen at the age of 16 or older. And if we look at young adults who were born prematurely, it's also seen that children who had a complicated start with a neonatal lung disease called bronchopulmonary dysplasia, they clearly have a lower lung function in young adults. So this is important, and it has important consequences potentially for the long-term health. But what are the mechanisms? If you're born too early, you leave the safe environment in the uterus too early, and your lungs and the body are exposed to the external environment that we all face, obviously, when we are born. But what are the mechanisms, and can epigenetics inform us about these mechanisms and long-term health consequences? That is what I will talk about in the remaining of the lecture. So what is epigenetics? Epigenetics refers to changes of the DNA of the DNA structure of the DNA code, but it's not the code itself, but it's chemical modifications of the DNA sequence. So if we look at this picture, methylation is one such change, a chemical modification of the DNA sequence. So here you have the C and the G, the base pair, the nucleotides. They are intact and they're not changed, but there is a chemical modification of the DNA sequence. Other epigenetic factors or changes are, for instance, histone modifications, how the DNA is wrapped in the cell. But for the sake of time, I will only focus on the DNA methylation today. So if we look at this picture, what does DNA methylation do in the cells and in the body? It has turned out it's a very powerful mechanism for the cells to regulate gene expression, meaning to turn off or turn on genes. And if you look at this lower picture here, where a gene is shown here in green, this gene can be fully transcribed and activated if there is no methylation taking place in the intron sequence or in the promoter sequence of this particular gene. Whereas if you see here above, there is lots of methylation sites that have occurred here in the promoter, and that could effectively suppress or actually shut down the gene expression in that particular cell. And it has been also shown recently that many of the environmental factors of relevance for health and disease, and perhaps particularly for asthma, allergy, influence the methylation levels and the methylation state in key genes and in key cells. So for instance, air pollutants may induce changes in certain genes. Aging is a very well-known factor that influence methylation levels. Factors like smoking, either active smoking or maternal smoking. I will get back to an example of that. Physiologic conditions such as BMI or traits or even obesity are linked to methylation changes. And then nutritional factors and dietary factors also influence methylation levels. And what can be the consequence of changes in methylation? Well, the concept is that it may certainly affect gene expression. And in this picture, you see here the famous agouti mouse experiment published many years ago now, where you observe a different color among these mice. And the only difference is that there is an epigenetic and DNA methylation change in the gene that regulates the color of the fur. And that in itself was determined also by nutritional factors. So it's a very powerful mechanism of the cell that can influence gene expression, that can also have phenotypic correlations, in this case, the color of the fur. I mentioned smoking, and in this particular study, we asked the question, if newborns and if children or fetuses in utero are exposed to maternal smoking, do we see methylation changes when they are newborn? And this is a consortium work with many thousands of children participating. This is what we call a Manhattan plot. It supposedly looks like Manhattan skyline with very high skyscraper, denoting the significance. So on the y-axis here is a minus log p-value, minus 10 to the log p-value. So here is a 10 to the minus 100. So many zeros before the value of significance, but certainly many zeros to indicate significance. And here are the chromosomes of the location of these particular DNA methylation sites. And as you can see here, there are numerous sites that correlate. Each dot here is one CpG site with a single methylation spot that is changed if the mothers were smoking during pregnancy. So there were hundreds of these sites, and we could also show that if there is a change in methylation, there is a change in gene expression. These effects seem to persist, at least up to school age. So if the mother was smoking during pregnancy, changes of the DNA were induced that we can detect in newborns, and these effects were persistent, at least up to school age. And the genes that were involved are known to be also correlated or associated with asthma, COPD, some cancers, neuropsychiatric diseases, and birth defects. So if we put this in a larger figure or in a schematic figure, we can say that maternal smoking during pregnancy has or leads to in utero exposure of the fetus. Then that induces these methylation changes in the DNA of the fetus and the newborn. And some of these changes influence gene expression. And now the big question, is it a causal pathway towards disease such as asthma? Also for other factors, I mentioned air pollution before, and we've done a lot of work trying to identify associations between air pollution exposure and these DNA methylation changes. And we also see these changes not as strong as maternal smoking, but we do see the changes, and also in genes that are relevant for lung disease and asthma. So this also highlights really the early life exposures that will have DNA methylation changes, and that could lead to gene expression changes. So what about asthma, and what about the disease associations? Are there differences if you have asthma, if you develop asthma, when it comes to DNA methylation changes? So for this project, we also collaborated with thousands with patients or children across Europe and the US. So several centers were participating. We analyzed DNA methylation with a chip called Illumina 450K chip, which means 450,000 DNA methylation sites analyzed. And we also really tried to replicate our findings to make sure we had robust findings, and then performed functional analysis and looked for gene expression signatures, for instance. And this paper came out last year, and as you see, a good number of authors were also involved in that. And if we look initially at whole blood, so we analyzed whole blood from children with asthma and compared whole blood DNA methylation signatures from healthy children, we could find 14 or 15 of these CpG sites to be differentially methylated, meaning there is a difference in the methylation sites. But if we look at this slide, it's actually quite small differences in the peripheral blood. And these are the asthmatics and these are the controls. Even though it was clearly significant and these could be replicated, the absolute levels were rather small actually. But when we then looked at isolated eosinophils, eosinophils are, as you may know, one of the key cells for asthma that initiates and maintains the inflammation in the airways, and also very linked to allergy responses. So here you can see the asthmatics and the controls. Now we see very dramatic changes in the DNA methylation level within the eosinophils. So that really pinpoints the role of eosinophils in asthma, and that DNA methylation levels can be one important factor behind asthma in children. I mentioned previously that there's a link between the methylation status and the gene expression. And we use this correlation also to annotate and really understand the findings that we were seeing with it when it comes to methylation. So we correlated the methylation levels and the gene expression levels in an online data set. And we can then see that the DNA methylation signatures that we identify to be different in asthma children and controls, they really correlated very strongly with gene expression levels that again highlighted the eosinophils. So these are typical eosinophil genes, and also CD4, CD8 T cells. And that gave us two important pieces of information. One is again that links us to eosinophils and the eosinophil activation in asthma, and also the T cells and the NK cells involved in the viral immune response in these children. And viruses are well known to trigger asthma and then could induce an asthma attack in children. So this is, we think, an important link between a DNA methylation and the viral infection leading to asthma triggering in children. So if we summarize these steps of the asthma epigenetics, we could see that many CpG sites, so single locations in the genome, were consistently associated with asthma across childhood up to the age of 16. We could not detect these differences at birth in this study, indicating that changes we saw here was acquired changes, so something happened during the early life of the children. And we noticed whole blood DNA methylation profiles, differences, but really driven by the lower methylation levels within the eosinophils. And this could be an epigenetic determinant of asthma. And if we look at the correlated gene expression signatures, we notice that there's a shift from the T cell population towards the effector memory cells, and that in itself is linked to host virus interaction for asthma inception. Another factor that's really strongly associated with DNA methylation levels or signatures is aging. And Steve Horvath's group has done a lot of research to really map the age differences when it comes to methylation. And there is now an online site that you can try if you want, and there is something called an epigenetic clock. So by taking a blood sample from a person, you can tell quite well how old he or she is just by determining the state of the epigenetic signatures. And we were interested in this concept that epigenetics can inform us about aging. And we also want to look at the early life effects and the preterm birth and birth weight. And this is one of the papers that looked at epigenetic analysis in newborns and the associations with birth weight, where a lot of signals really were seen early life. And this is a study that we are now finalizing epigenome-wide meta-analysis of blood DNA methylation in newborns and children, identifies numerous loci related to gestational age. And again, you can see this Manhattan plot. Here are the chromosome, and each dot is a CpG site and represent one methylation value. And again, here is the p-value, the minus log p-value here. Again, very, very significant changes. So there's strong correlation between the gestation age at birth and DNA methylation signals. And interestingly, if we compare the levels at birth, during childhood, at school age, and adolescence, some of the sites seem to undergo very rapid changes. So if we look at the first picture here, from birth to childhood and adolescence, you can see there is a major difference in the epigenetic level of this particular loci here. And for the next one, there's an increase of methylation level, starting perhaps at 0.3, 0.4 level here, reaching up to 0.6 or 0.7 almost, meaning that 70% of these CpG sites were methylated. Whereas for other of these CpG sites, there was a very stable methylation level, meaning that the level you are born with stays stable across childhood and adolescence. So the question is really here, is it epigenetic plasticity, so the changes with DNA methylation that can catch up and is a part of the development? Is it cell type issues, meaning that we're born with one set of cells that change during childhood? This needs to be investigated further now, really exploring those aspects. But it's a very fascinating research area, and we think these results are important. So my last part of the presentation, I'd like to focus forward a little bit. What's next, and what are we investigating time and effort now? And this is an ERC project that was launched a year or two ago, with the aim to target the childhood origin of chronic adult disease. I mentioned this concept in the beginning, and I'll wrap up with the same focus again. So we'll characterize the early chronic adult disease and try to predict outcome, and also hopefully one day prevent disease. So I'm involved in a large birth cohort called BAMSE. It started many years ago now, 1994, and repeatedly we've conducted questionnaires and clinical follow-ups with blood samples, lung function measurements, height, weight, blood pressure, etc. And we have now just completed a 24-year follow-up with a very extensive collection of data. So if we take the lung function map and the lung function trajectories again, here is from birth up to young adulthood. This is now what we are targeting at the age of 2024 in our cohort. We will focus on the low lung function growth trajectories and really try to identify factors and mechanisms involved here. And also, what can we detect the early biomarkers that could enable targeted intervention, really? In a bigger perspective, if we have a tough start in life, is there room for catch-up? Can we reach up to a good level here? This figure is a bit complex. Here are the supranormal persons with a very high lung function throughout life. The green again is the normal subjects with a normal lung function. Here the rapid early decline. And this is the group that had a tough start in life. But we are hoping also to focus on the catch-up. What can we help? How can we help patients and subjects who had a tough start with a low lung function to catch up and really achieve the maximum lung function in young adulthood? So if we go back to my patients that I meet in the clinic, I hope that with this research we will be able to say who should be targeted for disease intervention, who should have what treatment or an intervention effort. And overall, the ultimate goal here with the research is to provide new knowledge for targeted prevention in children at risk of adult chronic disease, and to identify potential targets for new asthma and COPD drugs. So to sum up, early life factors are important for later health and disease. And we know that impaired lung function in childhood is associated with chronic airway obstruction in adulthood. I hope I've shown that epigenetic and transcriptomic, meaning gene expression signatures, may give insight about the environmental exposures and also early disease mechanism. We have noticed very strong associations between maternal smoking during pregnancy and also other exposures or factors such as preterm birth and offspring DNA methylation. And we also find rather strong associations between epigenetic changes and diseases such as asthma. Going forward, really bridging the epidemiology with clinical and experimental work is really key, and I'd just want to highlight that and enforce those collaborations across disciplines. So with that, I'd like to thank you for your attention. This is my research group at Södersjukhuset, and I also want to thank the funding agencies for our studies. Thank you.